Hi everyone, the topic of our community project assignment is Duchenne muscular dystrophy in women, the diagnosis, cause, and available public supports. So the first thing that comes to mind when we read about Duchenne muscular dystrophy is how many of people, especially female carriers, show symptoms, and this number is only 8 to 20% of all DMD carriers. So essentially, Duchenne muscular dystrophy is an X-linked disease. Boys are whether affected or not, but females could be carriers. Dystrophin gene is a very large gene which is localized on location 21 of the P arm of the X chromosome. And due to the fact that dystrophin is a large gene and males have only one copy of X chromosome, there is a much higher chance for them to get DMD. However, there are females who get DMD as well, and the number of females diagnosed with DMD, although it's very tiny, and that's why many clinical trials and research studies have their main focus on males. And our main concern in this community project is to attract more scientific and public attention to the females with DMD and considering their challenges as much as males. Before that, let's take a look at the typical karyotype of a female with DMD. And interestingly, most of them show the phenotypes of Turner syndrome as well. And Turner syndrome is a disease specific to females in which one of the X chromosomes is missing or translocated to an autosome. Despite DMD, Turner is a common genetic disorder, and some of their phenotypes include a webbed neck and broad chest. The reason that scientists related DMD with Turner syndrome was different experiments in which they identified the chromosomal composition of DMD females and found that almost all of them had Turner syndrome. Specific genotype of all of them is 45X. Two main reasons that females get DMD is, first of all, the absence of X chromosome. As we know, females have two copies of X chromosomes, and in somatic cells of them, always one of these copies, whether from the copy from father or mother, randomly gets inactivated and is called the bare body. Now, if the good copy turns into bare body and lives off the bad copy in the cell, which has a mutation in dystrophin gene, then the female will get DMD. And the second reason could be that a part of all the X chromosome or all of it with dystrophin gene gets translocated to an autosome. And now we will read about some case studies. In this case study, Megan, Sarah, and Anna were born as triplets. And unfortunately, Megan and Sarah were both carriers, and it was thought that they would have a normal phenotype like their mother. But they were both slower to sit up and stand compared to Anna. Muscle biopsies then revealed that Megan and Sarah had low amounts of dystrophin, a symptom of DMD manifestation. Without treatment, Megan and Sarah sleep with braces strapped to their legs to stretch out their muscles. The problem is that the clinical trials for DMD are focused only on boys. Sarepta Therapeutics CEO was recently admitted that he did not consider about adding girls to the trials. Girls tend not to be included because they have variability in their dystrophin levels, and this makes hard to track improvements compared to boys who have no dystrophin at all. So how can we help? One is that we can help raise awareness about DMD in girls. We can change the names of campaigns such as Run for Our Sons to Run to End Duchenne. We can talk to friends, families, and co-workers about DMD and how women are affected as well. We can share our knowledge on social media. And two is to participate in researches on DMD in women. We can help funding DMD clinical trials on males and specifically on females, and we can participate in DMD clinical trials as healthy controls, especially if we are females. Thank you.